Good morning. So today we are starting our next unit on serology. So serology actually includes any type of bodily fluid that leaves your body. Okay, so we can take a look at saliva, semen, urine, blood, anything. Okay, this unit is also going to include DNA. So we're going to have two parts. It's a little bit longer of a unit, but we will have blood and then we'll also have DNA. So serology, just in general, remember anytime you see that ology, you know that it's the study of something. So when you see sera, you might think of serum, and serum thinks of like liquidy things. A serum, you might use a face serum or something like that at night. So serology, examination and analysis of any type of bodily fluids. All right, so when we look at blood in general is going to be that red thing that leaves your body, right? That's what most people say, okay, that's my blood. But specifically, when you look at blood, there's your fluid portion, which is the plasma. And you might know plasma because you can actually donate plasma and you'll actually get paid for that. Whereas if you donate blood, you do not get paid for blood, okay? The cells, there are different cell components within that fluid. The cells make up about 45% of your blood when you think of it. The technical term for a red blood cell is erythrocyte. They actually are responsible for distributing oxygen throughout the body. So if you know what a red blood cell looks like, it has a little concavity to it that allows for iron to bond to the oxygen as it comes in through our lungs, gets diffused into the blood, and then that gets taken to the rest of the body. So you might notice when you're working out, you breathe a lot harder because your lungs are breathing in, out, a little bit quicker because it's trying to get that oxygen diffused to all your muscles that you are using. The most important muscle in your body would be your heart. Leukocytes is the technical term for a white blood cell. They are responsible for cleaning for foreign invaders. They help with your immune response, making sure that they're able to help identify and then kill off any foreign invader that might be within your body. Thrombocytes are platelets, and they assist with blood clotting. So serum, again, is the liquid that separates from the blood when there is a clot formed. All right, so you guys probably know which type of blood you have. Either you are A, B, O, or AB. So the A, B, and O blood groups are class data or class evidence. So just because I am type B does not necessarily mean I am the person that committed the crime. You might have heard that you are B positive or B negative. That positive and negative refers to whether or not you have an Rh factor. So that Rh factor is what is found on those red blood cells. The Rh positive or negative is just whether or not you have a specific protein that is found on the surface of the red blood cells. Then you have antigens. This is what can actually stimulate the body to make antibodies. So certain antigens are found in the plasma of red blood cells, and they account for our blood type. And this will make a little bit more sense when we look at a picture here in a couple seconds. Antibodies are what are going to react with an antigen. So within your liquid portion of your blood, there are antibodies for the opposite blood types. So in case if you were to receive a blood transfusion, if you are type A and you receive blood from a B person, then your body would actually attack those red blood cells and they would clump and they would kill off those cells. The technical term for clumping is agglutination. So this is where it starts to make a little bit more sense. So you have blood type A, B, AB, and O. Hopefully you remember from biology, the blood type we receive is from our parents. So it's a combination of mom and dad. The alleles that you can see for type A, that would be IA. For B blood, it is IB. For AB is IA and IB. And you might note that there are two dominant alleles there. So hopefully you remember that that means it is an example of codominance, which means we will see both dominant traits. and I'll show you that here in a second. The only way to have type O blood is if you are recessive. 2 lowercase i's represent type O. So if I come back here and I look at type A and type B, there's 2 types of genotypes that can give me type A or type B blood. If I'm type A, I can be homozygous, which means I have the same 2 alleles, or I can be heterozygous for type A. But if you look, both of those have the dominant A allele. As long as the dominant A allele is there, that means you are going to exhibit that characteristic or that phenotype. Same deal with B. You can be homozygous type B, or you can be heterozygous type B. So that's how we would write it. Okay, so now let's look a little bit closer at those cells. So each of the red blood cells, you have type A, B, and O. The outside of those white blood cells have these antigens. Okay, if you look on the antigens, there's type A is on A blood. B antigens are on B red blood cells. And then again, because AB is codominant, you see both A and B antigens. Type O has 0 antigens. So remember I had mentioned that then within your blood, if I'm type A, I'm going to have anti-B antibodies in my plasma. So if you look, those would fit perfectly for something of that shape. So it would actually bond to those surface proteins, and then it would react and cause that agglutination to take place. But if you notice, that shape would not fit for our circular A antigens. Okay? You might notice since codominant AB has both antigens on its surface, it's not going to have any anti-A or anti-B, because if it did, it would clump to its own red blood cells and it would kill off its own cells, which is not a good thing. Type O, on the other hand, has both anti-A and anti-B antibodies, which means that if I give type B blood to somebody who's type O, it's going to react. Okay? So who can actually receive which blood types? So in case of an emergency, if you were to lose blood, okay, and you needed to receive a transfusion, type O is the universal donor, okay? Because if you look, the surface proteins, there are no antigens on its surface. So when you put that blood in somebody else, they are going to be able to assist with that oxygen transferring, okay, and transferring it to the rest of the body. The body will not recognize it as non-self and it won't reject it. Whereas if I look at AB is the universal recipient. So again, since it has no antibodies in the plasma, you can literally give somebody who's AB any type of blood. You can give them A blood, you can give them B blood, you can give them AB, and you can give them O. Okay? And then same deal with type A. You can see their compatibilities is A and O. Just type B is B and O. Now you would also need to consider Rh factors as well. If you are Rh positive or Rh negative, we would also have to consider that during blood transfusions. If you have a negative blood type, you can only receive blood from somebody else then who is negative, okay? Because your body would have antibodies against that positive Rh factor. But if you are a blood type that is A positive or B positive, AB positive, you can receive blood from anybody that would be a positive or negative, again, considering blood types as well. All right, so when you have a stain at a scene, there's a couple of things that have to get answered first. Obviously, number one, is that red stain blood? Is it human? Whose is it? Can you determine the blood type, any alcohol content? Do you have white blood cells present so you can actually identify DNA? Or is it just a blood type, which would just be class N, unfortunately? Okay. You can look to see how that blood might have been deposited. Was it due to a blunt force trauma? Was it passive dripping? or was it due to arterial spurts? So there's a couple presumptive tests that can help us quickly identify whether that substance may or may not be blood. Now remember, it is presumptive, so similar to what we did with our drugs labs, it was a presumptive test. It's not a confirmatory test. So there's the Kastle-Meyer test that allows for phenolphthalein and hydrogen peroxide causes a deep pink color if the sample did contain blood. You can actually use the hemosticks to actually look at blood, and it would react and turn a bluish green color. So if you have a water sample or a urine sample or something like that, you could test to see if there's blood in it as well. Luminol reacts with blood and it produces light. Luminol is probably the one that you think of in all the crime scene shows where they're using that black light and they're just shining it, and then all of a sudden all the fluids fluoresce. All right, so one of the questions we said we had to ask was whether or not it is human or animal. The easiest way to do this is take a look at a microscope. This test to see whether that blood reacts with another animal's blood. So the Perceptin test, this is kind of sad, they take the blood that they suspect may be human and they inject it into a rabbit. What they'll do is they'll take that blood, inject it into the rabbit, and then if the blood reacts, so the rabbit's blood is going to take it back out. If the antiserum is placed on our sample blood and it reacts with the human proteins, then we know that there is human blood present. Okay, this is very sensitive and it only requires a small amount of blood, which is good. Okay, when you compare animal blood to human blood, animal blood has larger nucleated red blood cells. Well, our red blood cells do not have nuclei in them. So number one, that's pretty obvious to be able to look at it and say, hey, it doesn't actually match up. Actually do. For us, remember, human blood cells, look at it. The slide looks completely different. We have very few white blood cells, and then we have lots and lots and lots of red blood cells. Those red blood cells do not contain those nuclei, and they're also a lot smaller. The other thing all right, so blood typing. Again, we know that our blood samples come from our parents. So mom and dad are both going to give each of their children an allele. So our alleles we are going to inherit are either going to be IA, IB, or lowercase I. Remember, the lowercase I represents our recessive allele, whereas IA and IB both are dominant alleles. Okay. We talked about which antigens and which surface proteins they actually have. We talked about who can they give their blood to, what groups they can actually receive blood from. Here's the distribution of our different blood types across the United States. One interesting thing is to ask your parents what blood type you have. If you don't have your blood type known, you can actually look and see, all right, well, is mine a pretty rare blood type? Because especially that AB positive, AB negative, those are pretty rare. So the Red Cross often needs a lot of people to donate in case of blood transfusions are required. Not only that, a lot of our universal donors, we should be donating often because then we can actually help save anybody. It does not necessarily have to be the exact same blood type. If I go back really quick, I just want to go over again how we can solve for what possible blood types our children may have. So let's say that mom has blood type A, and let's say that mom is homozygous A, and dad is type O. Okay, so there's mom, there's dad. Okay. The moment you read those two things, you should be able to generate your genotypes. So again, homozygous means it has the same two alleles, so that's IA, IA. And then dad is type O. Well, the only possible genotype for type O is two lowercase i's. I'm then going to set up a Punnett square. Hopefully you remember this from biology, flashback. And I'm going to set up my cross. Remember, the IA comes straight down, and then the lowercase I comes across, and you fill it in for the four possible outcomes of my offspring. So as I fill that in, I'm going to start to notice quickly that, hey, the only blood type that my children could possibly have is IA lowercase I, which the phenotype is type A, my genotype is IA lowercase I, and 100% of my offspring would have that exact same blood type. If I change it though, and instead of mom being homozygous, let's say that she is heterozygous, type A, and dad is still type O. So if I go back to this Punnett square, the one thing that is going to change now is instead of up here, instead of it being an I capital A, it's going to be a lowercase. It's going to be a lowercase I. So now here, it'll be lowercase I, and then lowercase I. So now the chance is that 50% of my offspring would be IA little I, and 50% would be type O, 2 lowercase i's. Remember, this is my genotype. My phenotype is what you see, so this would be type A, and this one would be type O. So if the possible offspring is a type B, well, hey, guess what? That doesn't match up. Maybe they're not the parents, or maybe one of them has the incorrect understanding of what their blood type may possibly be. So, all right, last thing that I want you guys to know is what a secretor is. A secretor is about 80% of our population. That means that their blood type antigens can actually be found in high concentrations across all their bodily fluids. So within saliva, semen, vaginal secretions, and gastric juices. Okay. All right, guys, that is it for today. You do have a quick little activity after this, so please make sure you go back onto Canvas and take a look at what you are completing for today. Hope you have a great one.